context for the studies is the problem of sepsis. Many of you may be familiar with this, and I'll just give a brief overview. It's defined as a life-threatening organ dysfunction caused by dysregulated host response to infection. And that sounds like a committee definition because it's long and kind of tedious, but it covers the very complex collection of signs and symptoms and problems related to infection that cause a very high mortality. The bottom graph summarizes a survey of different epidemiology studies that estimate the annual mortality to be between 250 and 500,000. The numbers vary depending on how strictly you define sepsis, but it clearly is a major problem. It's been studied extensively, and it's a fascinating case study of the complexity of the immune response to stresses and injury, in this case, infection. Normally, we'd like to have, after the onset of an injury, some inflammation, then dampening and gradual resolution, as indicated by the sort of dampening of this line. But in fact, in sepsis, there's often a systemic inflammatory response syndrome, cytokine storm, lots of exuberant active inflammation that leads to damage to target organs and multiple organ failure, part of the definition of sepsis. But it was also recognized over the years that in fact there's a second wave or an intermix of immunosuppression, which leads to secondary infection, and all that also leads to the complex multiple organ failure. And these two interact over time in these patients in a very complicated way. This has led to a frustrating search for novel host-directed therapies. We do have antibiotics for the infection and supportive care, and those two have improved over the last couple of decades to improve outcomes, but it's still a horrifically serious disorder. I put this up because part of the motivation for continuing to do transcriptomic studies in this disorder is the search for better understanding, which might, we hope, eventually lead to better therapeutics. There's been over 100 phase 2, phase 3 clinical trials of different targets that have failed. This is a poster child for why mouse studies can lead you astray, because most of these targets started off as working very well in mice but failed in people. We still need to make progress in this area. The other area besides therapeutics is diagnosis. And you might think this is rather simple, and in a perfect world, the patient does have things that tell you they're septic. They have fever, tachycardia, they have a likely source, maybe there's cloudy urine, they have laboratory tests or culture confirmation that lets you say, yes, they're infected. They have organ dysfunction, maybe their BUN creatinine in the kidney function department is going up, and it's a clear, easy diagnosis of sepsis that lets you intervene appropriately. But in fact, in many cases in the ICU, you have suppressed or absence of fever and tachycardia. The source of any infection or the presence of infection is not necessarily obvious, and oftentimes the conservative gamble is, well, let's cover with antibiotics just in case this person is septic. This actually leads to its own cost in terms of renal and liver toxicity with some of the antibiotics, cost, and other side effects. So there's a great need for a better set of biomarkers that lets us clearly say who is actually septic or not in these complex patients. So you might consider it, and I offer it to you that many of you probably aren't immunologists or working on sepsis, but it's a model system that's very challenging but representative of many chronic disorders where there's extreme heterogeneity in the cell types that are involved in the blood. There's person-to-person heterogeneity in their background and comorbidities. The time of samples is varied depending on when they get sick in their course in the ICU or elsewhere. The pathogens that cause the disorder are heterogeneous, and various treatments are being given to these patients depending on their comorbidities. And this complexity applies to other serious immune and genetic disorders. So if we make some progress here, we hope that it will apply elsewhere. Transcriptomics have been used to try to find sepsis biomarkers, and there's some encouraging hints. Here's a graph from a meta-analysis from Sweeney and co-workers. And you can see in this PCA plot that there is a fairly good separation with some overlap of patient samples with sepsis versus the non-septic inflammatory group, red versus blue. And in other components, there might be better separation. A problem has been that many different analyses have been offered lists of genes that might be useful, but there's actually very little concordance. There's about 8 or 10 different studies I've tabulated here, and some of them run off the slide. The colored ones show the small minority of genes that are found in more than one study, and most panels are unique. And it's still an open question whether any, and some of these groups are taking their subset into clinical trials to see if they're predictive. It's still an open question whether that'll happen and be successful or not. But there is a track record of trying to use this and eventually turning it into a PCR assay, as is possible with the DriverMap platform, which led us to get interested in trying our hand at this as well. So I'd like to share with you two sets of data about our efforts to work on this problem. The first is an ex vivo whole blood assay, and then subsequently some data from sepsis and ICU samples. The workflow uses the DriverMap protocol, which Gus has reviewed with you. I'll just mention, for my purposes, the key points, which for whole blood, which is the major currency of investigation in clinical research. Samples are easily obtained and don't require surgery, consent, risk of procedure, et cetera. We can look at total RNA without having to bother with globin depletion. Our ribosomal RNA is not an issue. Don't have to purify cells and introduce artifacts with the purification procedure. And if we make progress, this will be easily converted, given the nature of it, to a PCR assay. So all these are very attractive features. And so we took some normal adult whole blood, used the true culture system to stimulate the whole blood samples with endotoxin, the cell wall of gram-negative bacteria component that is very important in sepsis, and then applied DriverMap to look at the transcriptome responses to these samples. I do want to mention that among the other advantages of DriverMap, one thing that Gus mentioned in the y-axis on the left is the dynamic range, which is huge and a major advantage. And this comparison slide also lets me say that in our ex vivo data, we were able to compare the results with public data sets using either nanostring or microarrays to give us a sense of how the assay is working. And in fact, it worked quite well. I'll give you some summary of that before getting into the sepsis data. So if we look at the heat map on the left, the last column is the gene expression of these genes using the DriverMap. The other four columns are similar expression values from the public data sets deposited in GEO with their code up top. And broadly speaking, what we found when we did these comparisons is that we had greater sensitivity and range using the DriverMap on normal samples compared to the normal samples in these previously published and deposited data sets. One of them uses a nanostring protocol. And the comparison here also showed one other interesting feature, which made us think we were on the right track. Once again, we have on the heat map just two lanes now. Far left is the results from the published nanostring profile, showing a bunch of genes that are essentially down in nanostring, down-regulated. But you'll notice that there are a number, when the color scheme here is less bold than it should be, but there's about a top 10, let's say, of genes that we have a contradiction. The DriverMap actually shows them as up. And in that little box there on the right, if you go to the literature, there's data for 6 of them, and in all 6 of those 10, prior evidence says they're up with LPS. So we think the DriverMap is on the right track and is being more sensitive than in this comparison in any case. In the last version of this, if we look at all 400 differentially expressed genes reported by that nanostring study, whether you take average or median fold change, the direction was the same, but DriverMap generally gave you a greater fold change, and the median fold change was about 4 times greater with the DriverMap. So you get a better signal thanks to the dynamic range. So encouraged by that, we turned to trying to apply this to actual sepsis and ICU control samples. And in these early studies, here's what we have. This kind of relates to the question that came up with smaller numbers of samples. We have about 20 patients diagnosed with sepsis, 13 or so with adult respiratory distress syndrome, another reason to be in the ICU and be in dire straits, but you're not septic. Combination of the two, other people in the ICU that are there without these disorders, say with fractures or renal disease, et cetera, and healthy controls. So there's N here ranges from 13 to 24 per group. These were whole blood RNA samples. There's heterogeneity in the people, their age, the reason they're in the ICU, the time of obtaining the sample in their course, et cetera. And we have two kinds of controls. So we did the DriverMap profiling, found a whole bunch of differentially expressed genes, and in an initial exploratory data analysis, we did a standard approach, which is to look at variability with the PCA plot. And we could see that, thank goodness we could distinguish between normal healthy controls and everybody who's sick in the ICU. That's fine. But more importantly, the samples that were not septic were mostly separated from the sepsis with or without the additional component of ARDS, this adult respiratory distress syndrome. So this was encouraging. You can take the gene lists that come out of these profiling efforts and ask for sort of programs like Ingenuity, or in this case we use Metacore, another platform that does pathway and network analysis to give you enrichment analyses of what was inside that gene list. And if you notice, in the top processes, when we compared those that are differentially more found in the septic patients compared to the other sick people in the ICU, the distinction we're really interested in. Top hits here include immune system, response to stress, leukocyte activation, things that made biological sense, again, assuring us that we're sort of on the right track. And they also can provide enrichment with genes, enrichment values when analyzing patterns found in specific types of disease. And the top hit here for sepsis versus the controls was in fact bacterial infections and mycoses, again, consistent with the expectations and quite encouraging. We've also been able to identify gene sets of potential biological interest that we're going to be exploring. Here's one subset that has high expression in the sepsis groups, seen mostly on the right, compared to the control group in the ICU. And you can also use these tools to take a look at known networks that are highly enriched around key genes in your list. And in the Metacore symbol system, there's about 5 red circles here you can see. And those are the genes that were found by DriverMap. And the related genes allowed this system to build a network. And this is kind of approach that can get you started on a mechanistic analysis or look for new ideas in sepsis pathobiology for interest. Okay, so what I'd like to finish with is the question that we started with. It would be quite useful to develop a system to distinguish infected septic patients in the ICU from those that aren't. And are we on the right track? And so using these initial results, Gus turned to a support vector machine classifier methodology. And this uses a technique to separate groups. In A, it looks easy because we have a two-dimensional group B and A in a plane. To distinguish them is quite obvious, but the mathematics behind this actually uses a hyperplane and goes into multiple dimensions. And if you dare ask me a question about this, I'll refer you to Gus because that's the end of my knowledge of this approach. The product is very encouraging though. We were able, with cross-validation, to identify a set of gene expression markers that can distinguish the ICU control patients or the sepsis patients with about 85% accuracy, 88%. This is actually really extraordinarily encouraging for this relatively small number of patients. Of course, this has to be validated in future studies with larger numbers of samples, but it speaks to the power of this approach. Inside those markers that were used to generate the area under the curve graphs I just showed you, there are genes that you can, again, extract out for mechanistic studies as your interests require. So let me summarize. Thinking about the methodology, I can report from my perspective, the DriverMap is a terrific tool, has all the advantages of RNA-Seq and more, including generally it's faster, cheaper, easier, especially for whole blood. The idea of not having to manipulate it, that you can go straight from whole blood to your RNA analysis is a big advantage because we know and it's been demonstrated extensively by many groups, that any manipulation you do with your sample introduces gene expression changes that you don't really want to study, that those are part of the artifact. The rest of the methodology, applications, I've shared with you things I'm interested in, but certainly I'm sure you could think of applications for your own interests. In terms of the results, in our efforts to look at blood immune responses, we observed high sensitivity and specificity and a good dynamic range. We have encouraging results that show this is a promising approach for biomarker discovery and hopefully PCR conversion of those biomarkers for an assay that's clinically useful in sepsis and possibly in other immune disorders. I'd like to thank my colleague David Christiani, who provided many of the samples we studied from his sepsis studies, and Gus and Alex at Cellecta, who have been very helpful in driving this work forward. Thank you.